Welcome to part 1 of chapter 21, the immune system. We're looking at both the innate and adaptive immune system. It begins, as you can see, on page 764, and we're going to come over here to this page 765, and we're going to gaze momentarily at, you could say, what's clickable. And you'll notice that we begin with the innate system, and this is just a standard system for any multicellular organism that has a, a high-rise condominium of some trillions of cells, in our case 40 to 50 trillion. We have a skin covering, which we studied last semester, called the Integumentary System. We have a mucosal membrane wrapping through and, sealing off the oral cavity, the digestive cavity, respiratory cavity, and everything that has a lumen, including the heart cardiovascular system. Then we have internal defenses, phagocytes, specifically neutrophils and macrophages. We have also these special natural killer cells, which we'll talk about, very similar to a type of T cell that we'll study. We have inflammation. We have special proteins like complement. We have fever. Somebody asked me, what's the point of fever? And I wrote a little article about that in your recent emails. Then we'll press on to review what we've already studied, which is the adaptive defenses and the constellation of the macrophage, B cells and T cells that create humoral as well as cell-mediated immunity. So there's B cells and T cells involved in that. I would like to digress and just say that I was very fortunate after medical school to do a master's in immunology with the illustrious Dr. Remo Morelli, PhD out of Stanford University, whose postdoc work was involved in complement. So I got to have a really wonderful episode and group of experiences in graduate school in that particular area because of Dr. Morelli as my mentor. Let's take a look at, first of all, the innate system here. They start here with the innate defenses, okay? And what does that entail? And they give us some details here about that, having to do with the fact that we have an acid mantle on our skin. Interestingly, our skin does produce a fairly acid mantle when we perspire and so on. We actually secrete hydrochloric acid to some extent, or just acidity in general. Also, the luminous areas like the vaginal vestibule and the vagina itself is thought to have a normal pH that's a little bit less than neutral and wards off the, pathogens that might bother you when they're looking for a pH 7.4, which is what the interior of the human is. We have enzymes, lysozymes in general. We'll talk about that. It's part of phagocytosis. We have mucin, or mucous membranes, aspects of our saliva that trap particles. When we breathe, of course, we have the ability to trap particulate matter in the air, the inspired air, and sweep it out with cilia. We have defensins that we'll be talking about. There's a number of molecules that provide us with antibiotics or types of function. Okay, and then moving on from that, let's take a look here at this next page. We're on page 766, and I'd like to bring your attention particularly to the notion of sepsis. We are, as I've indicated to you, a high-rise condominium of 40 to 50 trillion congruent cells. We are, a clonal expansion of these cells. And within the confines of our body, not necessarily in the digestive or respiratory system, but, well, as soon as you get into the bloodstream and into the interstitial spaces, we are an antiseptic, or that is to say, we are how shall I say, purely ourselves, and we're sterile in that sense, which is to say we have our cells but no one else's cells. We have our products of cells and the products of no one else's cells. So when someone else's cell, like a bacterium or a fungus or some other type of thing like parasites for malaria and so on, enter us, then it's considered sepsis. Sepsis, most common forms would be where Streptococcus bacteria enter through the oral cavity openings and result in something akin to one of two types of results, which would be rheumatic fever or the type of facial features that result in a butterfly-like result on the face, a heating type of feverish result. Rheumatic fever is where cells actually end up in the heart and valve systems, and the neutrophils have to go after it, and they have a respiratory burst and they injure the heart valve. Rheumatic fever. So sepsis is always a very critical issue, and has to be dealt with aggressively in the medical profession. Barbecue sauce, we'll get to that. Meanwhile, phagocytes are what we're going to look at next here, and we have two general formats of the phagocytic persuasion, neutrophils and macrophages. Neutrophils, the first to arrive, the largest in number. Sometimes we call them polymorphonucleotides or nucleates because of their interestingly shaped nucleus. They are the first to arrive at every scene and respond very strongly to what's called opsonization, or we'll get to that in a second. Then macrophages are the last to arrive, and they deal with chronic issues. Actually, monocytes are, or macrophages, I should say, are monocytes which have escaped the bloodstream, and they are the last to arrive. You would think of granulomas with those. Sometimes they ingest so much particulate material that they sort of get a little, how shall I say, overloaded with digestive issues, and they end up getting cordoned off in the body. Tuberculosis would be an example of that, where the body actually uses connective tissues to cordon off areas that have gotten sort of mucked up with macrophage inclusion bodies. Okay, so let's look at the little buddies right now, and we talk about how do they know that they are needed. They have there's opsins out there. There's little molecules that get produced that have the effect of barbecue sauce, or if you've ever been in North Beach and you're standing outside of the pizza shops, I'm pretty sure that the kitchen actually pumps their exhaust fans onto the sidewalk in front of the pizza shop so that the passing by walk-in citizens of San Francisco smell the delicious smells of freshly baked pizza and find themselves entranced and sort of being tractor pulled into the enterprise for a piece of pizza. Anyways, that's the idea there, is that you want to encourage the neutrophils to arrive, and you want to paint some sort of a detectable scent onto what they're after. The process is called opsonization. When the neutrophils get there, they can do phagocytosis unless the object of interest is too large. They cannot surround the object properly, and they end up with what's referred to as respiratory burst, which is to say that in their compelling sense of duty, the neutrophils do not give up even though they cannot phagocytize something too large. Instead, they simply burst in place and release their bleach-like and digestive ingredients. And they have a little listing of here, liberation of highly destructive free radicals, oxidizing bleach-like substances, and basically very pH-rich acidic items and digestive material. So we'll pick it up now in part